Hello everyone and welcome. In this video we're checking out variant 1, variant 2 of October-November 2021. Before starting, make sure to check out the free notes I have available for you in the comment section down below. And don't forget to subscribe. Now let's go ahead and start with question number 1. There are two types of cell, prokaryotic and eukaryotic. Bacterial cells are prokaryotic and plant cells are eukaryotic. There are differences in the structural features that are common to bacterial cells and common plant cells. For example, the cell surface membrane in plant cells contains cholesterol, but in a bacterial cell the cell membrane contains molecules known as hopanoids, and these hopanoids are going to be carrying out the same function as cholesterol. Now, some of the main structural features common to both types of cells are shown in table 1.1, and complete table 1.1 by giving one difference between a bacterial cell and a plant cell for each structural feature listed, and then the difference between the cell surface membrane of the two types of cell has been completed for you. So here we've mentioned that the cell surface membrane of bacterial cell contains hopanoids, but for plant cells contains cholesterol. Ribosomes, for example, in bacterial cells are only 70S ribosomes, and 70S are the smaller sized ribosomes compared to 70S and 80S ribosomes in the plant cells. Now, right, 70S would be found in its mitochondria and chloroplasts, and 80S would be in its cytoplasm. For DNA, for bacterial cells, the DNA is going to be naked and circular and does not have histones, and it's going to be found free in the cytoplasm in the nucleoid. For plant cell, it's going to be found within the nucleus. It's going to be linear in chromosomes with histones, and it's going to be so it's not going to be naked. Okay, so that's the difference between the two. So here you can, obviously, because he said to give one difference, you can't give any more than one, and I really advise you to be strict on yourself here, because if you give more than one, you're probably gonna get a point marked off. So an example here we can just say no histones, and then here we can say, for example, histones, right? Another word for this would be naked DNA, and then here it would not be naked DNA. Okay, and then cell wall. Cell wall for the bacterial cell is made of peptidoglycan, which is a glycoprotein or a murine. And then I don't think I spelled it right, but anyway, plant cells, it is cellulose cell wall. One role of the cell surface membrane of bacterial cells and plant cells is the transport of substances into and out of cells. Explain how the membrane carrier proteins and channel proteins are involved in transport of substances into and out of the cell. Very simple. We know that the transport of substances in proteins is going to obviously be hydrophilic polar substances, ionic materials that are going to be obviously not capable of passing through the hydrophobic core of the plasma membrane. And so instead they choose the route that is easier for them, that is basically through the carrier proteins or the channel proteins, because the channel proteins have water-filled pores that allow it to be basically hydrophilic on the inside, and so do carrier proteins. Carrier proteins are also hydrophilic on the inside. But the difference between the two is that carrier proteins allow for transport of materials by two pathways, either by facilitated diffusion or by active transport, whereas for channel proteins it only allows for facilitated diffusion. So let's go ahead and write that down. Okay, so again, transport of ionic polar large molecules, well, it happens using carrier and channel proteins. And the reason for this is because these molecules can't cross the hydrophobic core or fatty acid core of the cell surface membrane or the phospholipid bilayer. So instead, they pass through these proteins. And you can mention here that channel proteins, because it's an extra point, you never lose marks by writing extra points. So channel proteins have water-filled pores, and the most important thing is to mention the different mechanisms of transport. Channel proteins allow for facilitated diffusion, but whereas carrier proteins allow for both facilitated diffusion and active transport. And here you can mention the definition of active transport for extra points. So define active transport. So see how you get marks here. Firstly, you write down the main points in transport of what materials. Then you write down here, all this stuff, this is extra points, okay? And then you would write down that channel proteins allow for what and carrier proteins allow for what. And if you want extra points, you will define active transport. You will write down that channel proteins have water-filled pores. You move on now to C is a photomicrograph showing chloroplasts in plant leaf cells. So you have chloroplasts. Explain why chloroplasts are seen only around the periphery or edge of each plant cell. It's very simple. In the plant cell, we have a central vacuole, and obviously it is not really shown because a vacuole it's single membraned, and I think this is a light microscope, so you can't really see the single membrane of the vacuole. So yes, there is a large central vacuole in obviously in the center if it's central, but the idea is that it pushes chloroplast as well as other organelles, as well as the nucleus to periphery. This brings me on to D. Figure 1.2 shows plant cells in a root tip where cell division by mitosis is taking place. Identify two cells in figure 1.2 that are different in stages of mitosis and draw a label line to each cell and add a name of stage of mitosis that is shown by the cell. Okay, so here we can label this immediately as metaphase, and now here I can see this is interphase, but obviously this is not a stage of mitosis, so I can't label. I can label this guy over here as prophase. I can also label this guy as prophase. It's really up to you, because you can see the nucleus is clearly gone. You don't have any nucleoli, you just have some condensed chromosomes. This is prophase, and there are two different stages, so I think we're good here. Okay. Identify two cells that are in different stages of mitosis and draw a label line to each cell. Okay, we're good. The high blood pressure at the arterial end of the capillary network results in some components of blood leaving the capillaries as forming tissue fluid at the venous end. The presence of plasma proteins allows for movement of water by osmosis back into the capillaries. Figure 2.1 in the diagram shows the capillary network with the lymph vessels and blood vessels at the arterial end and venous end of the network capillary are also shown. So you have the so here you could see the lymph vessels, and you can see the arterial end and the venous end of the capillary, and you can see the tissue fluid. Red blood cells and plasma proteins such as albumin remain in the capillaries and are not found in tissue fluid because they are simply too large. Explain why red blood cells and albumin do not leave the capillary. Exactly what I just said now. Red blood cells and albumin or plasma proteins in general, but here you mentioned albumin, are too large to leave, and thus they can't cross the endothelial pores, which are basically the pores in capillaries or between endothelial cells of capillaries. B. Name the chemical reagent or reagents used to test for proteins in a sample of blood plasma and state the color change that will be seen if protein is present. The only reagent that we know of is Biuret reagent, so write this down. Make sure to spell it right. Obviously this is not the correct spelling, but I'm asking you to spell it right. Color change is going to be from blue to purple if it's positive, as they have asked of you. Okay? This brings me on to C. When a person moves from sea level and stays at high altitude for a few months, there is an increase in the number of red blood cells. And I need you guys to memorize this, because sometimes they ask you to suggest questions about, like, if someone moves to high altitude, what happens to their red blood cell count? You have to know that it will increase, and this is an adaptation of the body, really. And if you think about it, it's very clear and very logical. Why? Because when you move into high altitude, when you live in, for example, high altitudes, I don't know, like wherever you live, like maybe if you start living on a high altitude, you'll find that when you first arrive there, you do not have, you feel like you don't have that much air around you, and so your heart starts beating faster, you start breathing faster as well, and so that's just to compensate for the lack of oxygen in the surroundings, and so for decreased hemoglobin saturation with oxygen, in the lungs. Okay, and obviously this results in less oxygen being supplied to the tissue, so that's why you're breathing faster, you want to supply more oxygen, your heart is beating faster, so that there's more supply of oxygen to the tissues. Now this is not healthy in the long run, so in the long run what we have is an increased number of red blood cells, so that, with the heart beating at the same rate, you just have more blood being pumped every second, because you have more blood. So why this increase occurs is because, again, at high altitudes, partial pressure of oxygen, you can also write it like this, okay? PO2 decreases. So basically, to compensate for decreased affinity of hemoglobin and obviously as a result of that decreased oxygen supply to tissue, more RBCs or red cells are produced to carry more hemoglobin, and this obviously results in more hemoglobin being available, so this results in just more oxygen being released to tissues, active tissues. D. A low blood albumin concentration can lead to a condition known as edema. Edema is a swelling of tissues caused by accumulation of tissue fluid surrounding the body cells and the capillary network. So just to explain how low blood albumin concentration can lead to edema. Okay, a low blood albumin concentration, I think here they mentioned something about this. They mentioned that plasma proteins allow water to move back by osmosis into the capillaries. Edema is going to be the condition where tissue fluid accumulates in the tissues. Obviously, that's the place where tissue fluid is. And so when this accumulation happens, this means that there's no drainage of the tissue fluid back into the venous end of the capillaries. And the reason for this is because there is no partial difference in osmotic potential between the tissue fluid and the capillaries. Usually water is going to leave at the arterial end because there's a really high pressure at the arterial end. Then it's going to come back at the venous end because there's a really high concentration inside the vein because of all of the proteins there and lack of water. So there's a really low water potential inside and a really high water potential outside, and so water is going to move by osmosis back into the venous end. Now imagine if there are no plasma proteins, the concentration in the venous end is going to be really low, which means that water is not going to move back and tissue fluid remains the same and it remains accumulated. And so that is it. Let's go ahead and write this down. So that's my suggestion. Okay. The low blood albumin concentration can lead to edema because low blood albumin concentration means that there is no difference in osmotic at the venous end of capillaries because no plasma proteins will mean that there is nothing to decrease water potential within the venous end. So water doesn't move back to capillaries. Obviously, water doesn't move back, and so what does this mean? Well, if water doesn't move back, there's no drainage of tissue fluid. And it accumulates causing edema. Now, obviously, if you want to go back to this question after you're done with the exam, you can write down the definition of osmosis. You can write down more reasons as to why there's no drainage. So you can mention, obviously, you can talk about how, at the arterial end, there is a high hydrostatic pressure, and so this is what pushes the water outside. At the venous end, you don't have that high hydrostatic pressure, so there's nothing to push water back inside unless there is osmosis, but this does not happen. Obviously, if you have time, go back and write down more. Now, albumin transports some cell signaling molecules from the cells where they are synthesized to their target cells. The cell signaling molecule bind to a specific cell surface membrane protein and on target cells. In the type of membrane proteins to which the cell signaling molecule binds, that is a receptor protein. Now, 3. Baculovirus is a virus that kills some of the insect pests of major crops. When the virus is released to the outside of the insect body, it's contained within stable protective structures known as polyhedrons. Okay, the main component of polyhedron is a protein molecule, polyhedron. These polyhedrons are sprayed onto plants as a bioinsecticide, and they're ingested by a feeding insect. And once inside the insect gut, they break down to release the virus. And so, explain why the term infectious disease can be used to describe the effect of baculovirus on insects. Well, in general, what's the definition of infectious disease? It's a disease caused by a pathogen that can be transmitted from one host to another. And so in this situation, we have a virus that is going to be the pathogen that is going to be transmitted from the plant to the insect. So just define infectious disease. So it's caused by pathogen, the baculovirus, and can be transmitted from one to another, or one host to another, whatever you'd like to say. It's just transmissible. And then suggest and explain conditions present in the insect gut that can cause the breakdown of polyhedrons. Okay, so I suppose in the gut there will be a different pH, just like our gut in the stomach, we have a really low pH compared to everything else. And that just means that if any protein was to come inside, that means it gets denatured because it's different to the optimal pH, since the ionic bonds are going to break and the hydrogen bonds break, so that breaks down the tertiary structure, and so it unfolds the protein, and that results in it changing shape, and so this releases the virus. I suppose that's the reason. Okay, so we can just mention that there is maybe a low pH or a different pH. It does not have to be low. In the human stomach there is a low pH. I suppose we can also suggest that it's a low or acidic pH, which denatures the enzyme. I'm sorry, not the enzyme. In this situation, it's just polyhedrons. Any protein will get denatured when placed in less ideal conditions. Yes? Okay, by breaking down ionic interactions, then hydrogen bonds, stabilizing, that used to stabilize, that stabilized the tertiary structure, which now breaks, unfolding the protein, releasing the virus. Now here we have a polyhedron molecule is composed of 3 identical polypeptides. Each polypeptide is 245 amino acids long. The first 10 amino acids of polypeptide are shown in figure 3.1. So you have methionine, proline, asparagine, okay. And in figure 3.2 is a ribbon diagram of a single polypeptide, and figure 3.2b shows a polyhedron molecule composed of 3 polypeptides. So you have the entire polyhedron molecule versus a single subunit of the polyhedron molecule. And he's saying here, with reference to figure 3.1 and figure 3.2, describe the structure of polyhedron. Okay, so in general, 3.1 and 3.2, here you can see the primary structure of polyhedron with its amino acid sequence. Here you can see the secondary structure of polyhedron with the alpha helices and beta pleated sheets. Here you can see the coiling also, so you can also see the tertiary structure of a polyhedron. And then you can see the quaternary structure with more than one polypeptide chain being involved. So that's all, okay, and it has 4 marks, I think each mark pertaining to a different kind of structure. So you have a primary structure in figure 3.1, which is the definition, amino acid sequence, and it involves which kinds of bonds? Peptide bonds. So remember, in every structure, mention its definition and mention the bonds that stabilize, okay? There's also what's it called, a secondary structure, in figure 3.2 with alpha helices and beta pleated sheets. What kinds of bonds stabilize them? Stabilized by hydrogen bonds, a whole bunch of hydrogen bonds. And then after that you can mention tertiary structure. And what is tertiary structure? Well, it's actually the coiling and folding of protein maintained. I mentioned the different kinds of bonds. Obviously, right, could, because not all bonds are involved. So could be maintained by hydrogen, ionic interactions. Okay, don't forget bonds here. Ionic interactions, hydrophobic interactions, disulfide interactions. And then you're done with that. Okay, quaternary structure in B. Okay. Yes, I think it was figure B. With more than one polypeptide chain. And that's all. This brings me on to D. The sequence of DNA nucleotides in the gene that codes for the polyhedron polypeptide has been determined. Okay, and he's saying he explained why the amino acid sequence of polypeptide cannot be used to deduce this sequence of nucleotides in the gene that codes for polyhedron polypeptide. Okay, the reason that the amino acid sequence cannot be used is because the genetic code states that H3 nucleotides code for a single amino acid, which means since we have 20 amino acids, we have 64 codons. Okay, so there's obviously a bunch more codons that there are amino acids, which means that we have like 2 or 3 codons for a single amino acid, and so obviously you can't have a way to determine like, oh, which codon is going to be the one in the gene. Besides all that, don't forget that in a gene you could have exons, could have a coding part, you could have introns, you could have a non-coding part, which means that overall you're not going to have, the perfect time deducing the nucleotide sequence. Okay, so again, first make sure in any question, any question about what's it called, genes, and about amino acids and codons, genetic code. Okay, it's the first thing that you write down. The genetic code, basically each three each 3 nucleotides code for one amino acid, and we have 4 nucleotides, 64 codons, and only 20 amino acids, which means you have 3 or 2 codons, even more sometimes, per amino acid. So obviously you have no way of determining which per same amino acid, determining which is used in the gene. And there are introns, non-coding parts of gene that also couldn't be determined. Using amino acid sequence. Now this moves me on to 4. Berlinka is a variety of common grapevine, Vitis vinifera or something, and Berlinka grapes are used for making wine and are sold as fruit. There are economic and glycological benefits from using less water to irrigate grapevines, and you are using high crop yield. The rate of flow of sap within xylem vessels and roots to leaves can be used as an estimate of the rate of transpiration. This also indicates water uptake. And he's saying here the hydrogen bonding of water molecules is important in the transport of sap within xylem vessels. See the term used to describe molecules sticking together in the xylem vessel. Together is cohesion. Water molecules sticking to the cellulose molecules is adhesion. And then you have an experiment was carried out to investigate the effect of leaf area on the rate of flow of xylem sap during the fruit development. The flow rate was measured over a 3-day period in 3 Berlinka grapevines. with totally different surface areas. And he's saying here that the plant was grown in the same conditions. The difference would be that the leaf area that was 13.7, 9.6, or 3.2, and the one that was lined is the smallest. So this line over here is the smallest, and you can see the flow rate in xylem is a whole bunch less compared to this. And I think this makes sense because here you have more transpiration in the one with the bigger surface area. So this means that there's a greater transpiration pull and overall more water flow compared to the last one. Now he's saying here that the general pattern of results in figure 4.1 is the same for 3 Berlin cup grapevines. Explain why the general pattern results is the same. So why the general pattern overall is the same? Why do we have an increase in this time of the day and then a decrease in this time of the day? Well, I suppose again because during the day the stomata are open, there's more transpiration, and because they're all placed in the same conditions, they're all affected the same way by the change in the weather and the change in the environment. Okay, so let's go ahead and write that down. Okay, so that's an explanation, I suppose, again, because during the day stomata are open, and obviously the stomata are open, so there's more transpiration, and that just results in more water flow in xylem. And obviously they're all going to be affected the same since they are all placed in similar conditions. Actually, they're all placed in the same conditions. Now this brings me on to 2. With the reference to figure 4.1, explain the difference in the results between the 3 grapevines 1, 2, and 3. So again, why does what's it called leaf area grapevine 1 have the greatest amount of flow rate compared to the 3 with the least amount? And this just has to do with surface area. Here, because they have the greatest leaf area obviously contributes to surface area. It contributes to having more stomata, it contributes to having more transpiration and more gas exchange and everything, and so that just results in, if there's more transpiration, there is a decreased pressure at the top, and so that just causes the xylem flow to just increase, right? So that's the reason. Okay, again, for grape, not like describing, just like go ahead and start explaining. Again, you just say here that greater leaf area means greater surface area, which means transpiration rate increases. More. So that's transpiration rate. You have another thing called so there's transpiration rate, and there's also more stomata, and that also increases transpiration. Okay, so transpiration also increases because of that. And then you can mention another thing, which is when there's a greater transpiration, contributes to the transpiration pull. And flow of water in xylem. Suggest an explanation for decrease in flow rate in xylem sap shown at time p in figure 4.1. Time p. So all of a sudden there's a decrease for all of them on day 3. This could be anything that can affect transpiration. Maybe the weather changed, maybe like it became more humid or became rainy, or maybe the wind speed changed, decreased, maybe, anything that you can think of that changes the rate of transpiration, just write it down. But he just wants an explanation, so just an explanation, I suppose. Maybe weather changed, became more humid or rainy, and that decreasing the rate of transpiration. Okay, this brings me on to 4, outline how you would determine the surface area of one side of a leaf. I would use the grid. Okay, place the leaf on a grid, and this grid should have, squares on it, so equally divided area, of squares. And then I would count the number of squares that this leaf would fill up overall, and then I would multiply it by the area of a single square. Obviously, if the square was already 1 centimeter square, then I would not have to multiply by the area of the square. Okay, so place leaf on a grid which was divided into squares. Of equal area, and count the number of squares, and I think this skill you'll be able to gain in the lab as well. Yes, count the number of squares filled by the leaf. As for this question over here, it's a 2-mark question, but I feel like you should be able to fill more information here. So greater leaf area means greater surface area, transpiration rate will increase, and obviously more stomata are going to be open, so also transpiration will increase, and a greater transpiration contributes to the transpiration pull and flow of water in xylem. But again, you can write down more information. I know that this is only an explain question. You can still compare. You can still, for example, compare this 1 to 2 to 3. You will never lose, grades if you do that. It's again just a 2-mark question, but to be safe, if you have time, go back and fill. And then the causative organism in measles is morbillivirus. Young children who have not been vaccinated for measles are at high risk of becoming ill and developing complications associated with the disease. The genetic material for morbillivirus is a strand of RNA, and statements of ABC relate to the structure of RNA. State the correct term that matches each of statements A, B, C, and then the name of 2 purines and 2 pyrimidines in RNA. Okay, so for purines, it's adenine and guanine. Make sure to write down the full names. You can't just go with A or G, okay? Pyrimidines, obviously make sure to not write down thymine because there's no thymine in RNA. It's going to be cytosine and uracil. Now for the type of covalent bond between RNA nucleotides, it's a phosphodiester bond. And then the pentose sugar of RNA, it is ribose, which is why it's called ribonucleic acid. And then describe how measles is transmitted. Okay, measles is transmitted basically through, it's an airborne infection just like tuberculosis, and I think measles is no longer part of your syllabus. I just think that you still should know, the what's it called, a basic method of transmission. So it is airborne, just like, how you would talk about tuberculosis, you would talk about measles. It's airborne when someone, it is host, infected with measles, coughs. Obviously their coughs, he will release air droplets, what's it called, which will contain the pathogen. Which could be inhaled by someone else. And don't forget, it is a transmissible disease, so this is all it takes: a pathogen, a host, and someone to transmit the disease. Okay, so by someone else and when inhaled, the measles goes into the system, and that's all it takes. Obviously it's a 3-mark question, so make sure to write down as much as you could, just about the same thing. You don't have anything else to write down. If you wrote it's airborne and you write host and you basically talk about the air droplets and inhaled by someone else, you're good to go. But I would keep on writing stuff until the lines fill out. C. Young children who have measles have develop difficulties with breathing. This is made worse if the child is continuously exposed to tobacco smoke, and tobacco smoke contains harmful compounds such as carbon monoxide. I know that smoking is no longer part of the syllabus, however, we're going to make sure to check this. Okay. Inhaled tobacco smoke passes through the larynx, which is the voice box, and other structures in the gas exchange system in the alveoli. So this is not a question about, smoking. It's a question about the gas exchange system. Complete figure 5.1 to list the correct sequences of the main structures of the gas exchange system through which tobacco smoke passes and to reach the alveoli. So from larynx to alveoli. Above larynx, we have the pharynx. So the pharynx is going to be the throat, and then the larynx is going to be your voice box. Below the larynx is going to be the trachea. So we write down here trachea. Trachea then splits into 2 bronchi, right and left. After that, they start to split into bronchioles. Okay, so bronchioles. Okay, so main bronchi, and then more bronchi, and then in the end you're gonna have bronchioles, which then split into alveoli. State the short-term effects of carbon monoxide on cardiovascular system. What we know about carbon monoxide is that carbon monoxide has a really high affinity to hemoglobin, actually a greater affinity to hemoglobin than oxygen does. So this means that if carbon monoxide was inhaled, it's going to stick to hemoglobin, preventing the as basically movement of oxygen in the body, preventing the transmission of oxygen to the body tissues, and so this obviously results in specifically on the cardiovascular system, in the oxygenated blood, and blood not being, blood not carrying oxygen at any time being. So let's write this down. Carbon monoxide has a really high affinity to hemoglobin, greater affinity than oxygen, and it binds to hemoglobin, forming carboxy hemoglobin, and this results in deoxygenated blood. Okay, the iPad pencil turned off. Okay, yes. Resulting in oxygenated blood. This brings me on to D. Smallpox is a disease that has not been yet eradicated with the help of global vaccination program. Measles has not been eradicated even though there is a global vaccination. Okay, so there is a global vaccination. Measles has not been eradicated, but smallpox has been eradicated. He's saying here this is a graph showing the estimates of number of deaths from measles globally between 2000 and 2017 for people who have not been vaccinated. And you can see the number of deaths per million, and then over years you can see that the deaths per million keep increasing for people that are not being vaccinated, which obviously means that there are more people not being vaccinated. And so he's saying state the trend shown between 2000 and 2017. Between 2000, so from the beginning till the end, you can see there's a constant increase in the number of deaths from measles between the years 2000 and 2017. And since this is just a one mark question, I think just writing down this trend, as in there's a steady increase in number of deaths per million, basically in million, not per million. Okay, in million. Yeah, so a steady increase between the years 2000 and 2017. Okay, and yeah, so this should get you the mark. Okay, so just reasons for the trend shown in figure 5.2. So the reason for the increase, I would suppose, could have to do with obviously the number of unvaccinated people is increasing, but like overall, as the years pass by, there'll be more people, and there's more people that are unvaccinated as a result of that. And don't forget, when it comes to vaccination, the most important bit is herd immunity. Herd immunity is the idea that if most people in the community or population are vaccinated, this means that the spread is going to decrease by a whole bunch, because for every infected person, 5 more will not be infected. And so the idea here is that everyone is safe and protected. Now, in a situation that there is no herd immunity, this is going to result in an increase and no eradication of the disease. Another reason could be that there's a rise of people who are basically against vaccination, and basically there's the anti-vax communities who are all against vaccination due to beliefs or cultures or religion, whatever it is. It could be a reason for no vaccination, and they do not vaccinate their children, so more and more no herd immunity. And let's not forget other reasons for increase in the trends shown. I think another reason might be that maybe the vaccine program itself is expensive, or that the vaccine itself, it can't be maintained properly. So usually there'll be a problem in the vaccine if it has to be maybe maintained within a specific temperature at all times. Like the Bacillus calmette-guerin vaccine for tuberculosis, it also is like this. And so I think vaccines are like that. They're expensive and they're hard to maintain just the same. So I can just write these all down as reasons. So again, there is so there's no herd immunity. And greater, there's obviously an increased number of people per year. With more people being unvaccinated, and reason for this is again maybe there are people against vaccines. And why are they against vaccines? It's very simple. It could be just belief or culture. They don't vaccinate their children. They themselves are not vaccinated. So again, that contributes to no herd immunity. And then besides that, you can mention, they don't get vaccinated. Vaccination program might be expensive. Vaccine hard to maintain. What else could we say? We can write down extra points here about how if people don't get vaccinated, they don't get a primary immunity, which means they don't have a fast secondary immune response, which obviously results in a greater number of deaths. Now, a child with leukemia is at high risk of developing measles even though the child has a high white blood cell count and has been vaccinated against the disease. To explain why this child is at high risk of developing measles. Okay, so despite them having a high, white blood cell count, this does not mean necessarily that they have a high immunity, because in the end, they just have, obviously cancer means there's uncontrollable cell division of white blood cells. It could be of any white blood cell. It doesn't necessarily mean that they have a secondary immune response. Even though they said that they've been vaccinated, the child with leukemia has a weakened immune system because of the cancer, because they have these undifferentiated cells that are just basically, continuously dividing. The other cells are not dividing, the cells are not, actively responding to the disease, they're not actively responding to the situation, there's no memory cells produced, and there's basically, it's basically like an opportunistic infection, just like if someone was to have like maybe HIV or AIDS. This means that they're going to have a weakened immune system, and overall, like, any disease is going to be, hurting them. Any disease, like, it's, an opportunity for any disease to invade their body. Same situation, I think, with leukemia. So with leukemia, now, with questions like this, where like you don't necessarily know the answer because you never like dealt with a situation like this, just make sure to write down everything that you know. Any keyword that you might think pops up in your mind, just write it down. Okay, so no primary immune response. Therefore, no, active immunity. And then here you can write down that it might be an opportunistic infection. Since leukemia decreases person's or child's immunity. Okay, now this brings me on to 6. Amylase is an enzyme that catalyzes the hydrolysis of starch into reducing sugars. Okay, students carried out an experiment to investigate hydrolysis of starch using immobilized amylase, and figure 6.1 is a diagram of the apparatus that was used in the investigation. So you have a starch solution, and the starch solution here is going to be released into a immobilized amylase in alginate beads, and then you're going to have a tap and a beaker for collecting the product, which is going to be starch broken down into reducing sugars, which are going to be alpha glucose. Now this brings me on to the alginate beads were the same size, or maltose, maltose. So yes, the alginate beads were all the same size, and he's saying that both taps were open to allow starch solution to flow down the column for the product to be collected, and the product was tested for the presence of reducing sugar and starch. The results of the investigation showed that the product collected in the beaker contained reducing sugar and starch naturally, because not all of it had the time to break down. And so with reference to figure 6.1, describe a method that would allow students to use the immobilized enzyme to collect only reducing sugar. So to make sure that you collect only reducing sugar, you could add more alginate beads, which means that you can basically maybe use smaller alginate beads and use a whole bunch more, because first of all, when you use smaller alginate beads, this increases the surface area of the enzyme, and so less of the enzyme is covered by the alginate bead, and the active site is more exposed to starch, so it could be broken down. Plus you're using more enzymes, so overall there's a higher rate of formation of enzyme substrate complex. You can also maybe I suppose you can keep it in there for longer, open the tap slowly, only release one droplet at a time, so that you don't pour it all in at once. Open the tap slowly, release a small amount, and then continue on with the work. Don't open this tap also so quickly, leave it in there for a while, I suppose. So yeah, that's what I would say. Again, use smaller enzymes and alginate beads. Or what do they really call them? They call them alginate beads. Okay, and more of them to allow more enzyme, to allow the enzyme to interact more with substrate. And again, leave starch in, what's it called, the column longer before opening top to release. And release starch slowly. So basically, okay, now one standardized variable in the investigation is the size of alginate beads. He states just one reason why using larger or smaller alginate beads in the column would affect the results obtained. That's exactly what we said here, because again, if you use smaller or larger, that just changes the surface area. The smaller the bead itself, the greater the surface area to volume ratio, the greater basically the interaction between the substrate and the enzyme, and the more the formation of the enzyme substrate complex. So again, smaller have greater Now this brings me on to 16, and that's all for this paper. Now I hope this video was useful for you. If it was, make sure to like, share, and subscribe, and I'm gonna see you guys in the next one.